is I am a researcher at Jimma Agricultural Research Center, and currently I'm a coordinator of Biotechnology, Plant Biotechnology Department. To begin with the lab organization, like any other tissue culture, we have washing room, media preparation room, culture, media and equipment sterilization room, and also we have aseptic transfer room and plant growth chamber or growth room. Thank you.
My name is Elias Gauramanam. I'm a researcher at Jimma Agricultural Research Center, Plant Biotechnology Laboratory. Currently, we are doing coffee somatic embryogenesis using 3 protocols, Van Boogstein and Bertollet, the first one. The second one is Texera and Etad, 2004. And the third one is JARC protocol. So in the following video, consecutive video, I shall demonstrate 3 steps. The first one is expand sterilization, on which we use sodium hypochlorite, ethanol, and 320. And then after cleaning the expanders, we directly transfer it to primary callus initiation media. Then we will keep it for 30 days. Then after we will transfer it to the embryogenic callus induction media. So here we identify embryogenic callus using morphological only. Therefore, our major problem in this case is that we have 2 problems. The first one is identification of embryogenic callus using histologically. Well, morphologically we identify, but histologically we have technical problems. The second technical problem is after obtaining such embryogenic callus, then suspension culture, establishing suspension culture is our second major problem. So because of this, we directly transfer embryogenic callus directly to regeneration media by skipping multiplication media. So the next step is elaborated by my colleague. Thank you.
I'm going to demonstrate the regeneration steps. Under this activity, we will identify the embryogenic callus from this jar. The embryogenic callus development is clearly observed when the callus turned into yellowish or sometimes brown colors. After that, we take this embryogenic callus and transfer this embryogenic callus into the regeneration media. After certain period of times, as the embryo regenerated, we will count the number of embryo per explant. Thank you. Okay, thank you. I'm going to demonstrate you the embryo regeneration and development step. In this step, from the previous step, we identified the embryogenic callus, and we take this embryogenic callus and directly transfer this embryogenic callus onto the regeneration media by skipping tissue multiplication or proliferation step. In coffee somatic embryogenesis, getting the right embryonic tissue, embryonic callus is the most critical step, especially in direct somatic embryonic pathway. In this jar, you can easily see that these callus are already turned into embryo, or they are regenerated into embryo globular stage under different experimental, under different media composition or experimental setup. However, sometimes we misidentify the right embryogenic callus. Due to that, these embryonic callus are unable to regenerate into embryo. So this is the major problem that we are facing in our lab, and we are looking forward to have some technical backup on this. After the embryo regenerated, we will proceed into the embryo development for maturation state. So this is in this chart you can easily see that some of the embryos have already changed into cotyledonary stage. So in most cases, and in these embryos has to be transplanted or planted in the nursery. However, due to the poor greenhouse facility, we do not perform this activity. Rather, we acclimatize our embryo in vitro, under in vitro condition. As you can see from this chart, these are the somatic plantlets which are being acclimatized in the lab. Thank you.
This is our growth room. This growth room contains light as well as temperature control. This temperature controls will be 25 plus or minus 2, which is standard with environmental temperature, as well as the lights will be measured by lux, and 2000 lux will be there for photosynthesis purpose. And as you observe in this, plantlet will be growth from the embryo. These embryos will be torpedo stage and cotyledonary stage. Those cotyledonary stages will be growth after 2 months to plantlets. Those plantlets having contains a lot of growth system. Those growth systems was consisting of root as well as shoot, even if also stems. So these plants will be already ready to acclimatizing. So those plants will be acclimatized at the greenhouse. In this laboratory, we have 2 problems. Those problems will be, one is contamination, another is necrosis. Contaminations may be caused through the microorganisms, maybe fungal as well as bacterial. Those contamination will be isolated through observation. Another is defoliated. Those defoliateds will be maybe causes for mineral nutrient as well as agent, and this necrosis, as you observe in this video, the necrosis will be there, defoliates also there. Those problems cannot be solved by our capacity. This is primary acclimatization of coffee. This, as you observe in those seedlings, which are very beautiful, and they control through the cold treatment. And this, there is a lot of problems. One problems will be defoliated as well as dumping off. So these problems will be solved through antifungal or other treatments. As you observe in this greenhouse will not automatically, this is manual. So there are a lot of problems. If you will modernize this greenhouse, those problems will be solved easily. This is greenhouse. It doesn't say modern greenhouse. This is net house. So these net houses will be acclimatized for coffee in secondary acclimatized. So the secondary acclimatized will be there. So those coffees will be good and easily distributed to our farmers. So in this problem, there is a lot of problems in this. Those greenhouse will be not automatically functional. We are always treated with manual practice. And then this net houses was very difficult to managing heavy rains as well as many insects. So these insects as well as microorganisms will easily proliferated or easily damaged our seedlings. So these problems. If these problems will be solved, we are escaping to optimizing our acclimatizations of coffee seedlings.